excited to see what you're doing. And everybody, please give her a round of applause, and we'll kick off our last FeMS Mixer of 2020.
Awesome. I can do that. Everybody can see this, right?
Yep.
Cool. Let's get a pointer going. All right. So, hi, everyone. My name is Devin Swiner. I'm a 5th year in analytical chemistry at The Ohio State University. And I'm just going to kind of talk a little bit about some of the work, like very briefly, that I have been working on so far here in Dr. Abraham Badu-Tawia's group. So just a little bit about me before we kind of jump into my work. I am from Maryland, so I'm an East Coast girl. I was born and raised in Upper Marlboro. I'm a graduate from Pitt, so I went to Pitt and graduated in 2016. I got my BS in chemistry, but I also have minors in sociology and Africana studies. While I was at Pitt, I did research under Dr. Tara Meyer. So I did polymer research. I don't work with polymers anymore, but I was a baby analytical chemist. So a part of Tara's group synthesized block copolymers with lactic and glycolic acid monomers. And it was my job to pretty much just play with them and see how, the sequence of those polymers affected their mechanical properties, and then try to extend that to various applications. So now I'm at Ohio State, and I basically work on mass spec-based method development for clinical diagnostics. Some other fun facts. I love giving a good fun fact slide. I am one of the Black and Chem co-founders. So that launched on Twitter this past August, which was super great to watch unfold in real time. I am a self-proclaimed MC Hammer's Twitter best friend. We follow each other on Twitter. I can tweet him, he tweets me back. I did a book giveaway that he retweeted for me and everything. So I feel like by social media standards, that means we're best friends. I'm a huge STEM advocate, so other things, I do tend to wear many hats. I'm currently the chapter president of our Nobuche chapter, and if you're not familiar with Nobuche, it is a nonprofit organization that caters to Black chemists and chemical engineers. So some cities have professional chapters, but a lot of universities have student chapters. So I'm president of our student chapter. It's going on, I think, my 2nd year as president. This is just a picture of some of us at our annual Nobuche conference here. A lot of the work that I have been doing in Nobuche got me an award in 2018 for mentorship and leadership, which was really awesome. I'm also a founder of Max Sciences, which is just a blog that is really for women of color in STEM. So we are really candid, blogging about our journeys and what's worked for us, what's been great, our ups, our downs, everything in between. And the work that I've been doing there also got me an award from the WCC for encouraging women into the chemical sciences. And then finally, another fun fact, I'm a huge science communicator. So anyone that on here that follows me on Twitter knows I'm always have threads that are filled with GIFs. Like that is like my claim to fame, which is what I really love doing. And you can follow all of that on my professional website, so devinthechemist.com. But into the research. So my work is really focused on clinical diagnostics. So let's say you have a clinical sample and you can do one of two things with those. You can either do point of care testing. So think about like diabetes testing where you do a finger prick and check your glucose levels, or like a pregnancy test. These are point of care testing methods that you do at your home. Or you could take it to a centralized laboratory. So this mimics like going to the doctor, getting your blood drawn. But for both of these methods you can take with the clinical sample, you can do direct analysis with. So you want to take that sample, actually analyze it, and then get out some form of result. Where it differs a little bit is for point of care testing, you want to figure out how can we make this miniature? How can we ship it, or how can we put it in the store for people to buy so they can actually use? Whereas for a centralized laboratory, you're thinking more automation, how fast can we do this analysis, or even multiplexing, whereas like, can you take one sample and get a lot of testing out of it? But regardless, whether you're thinking of point of care testing or centralized laboratory, you're trying to reduce the turnaround time. So that's the time it takes from getting the sample to actually analyzing the sample to giving you results, because the quicker that happens, the easier it is to put in place certain protocols for treatment. And because of that, that also improves engagement between the clinician or the doctor and the patient. So what my work has been focused on is this direct analysis step for both a point of care test and also for a centralized laboratory. So when we think about this, typically for clinical diagnostics, you might think of a very traditional LC-MS. So you're taking your sample, you're doing some form of sample prep, then you're separating it out, getting your mass spectrum out, or if you're doing UV detection and getting your results. And we know that for LC, you can couple that to mass spec via electrospray or like atmospheric pressure chemical ionization, so APCI. But another way that you can couple clinical diagnostics to mass spectrometry is through ambient ionization. And that is what my group is really focused on. And if you're not familiar with ambient ionization techniques, these are a class of techniques where we do the sampling and the ionization outside of the mass spec. So there are a lot of different sources for ionizing agents. So for DESI, for example, you're using solutions and a nebulizing gas to do desorption on some form of solid sample. Then the ionization step is after that. You can also use plasma, lasers, etc. But what's really great about these ambient methods is that they're compatible with raw sample analysis. So we don't have to do any sample preparation, which, as you can see here, is the bulk of the time for LC or even GCMS methodologies. So the mass spec is the short part regardless, but with ambient ionization, we can do all of these things really simply and also very quickly. Now, a subclass of ambient ionization are substrate-based methods. So that's what I have been doing. So if you think about it, you can take any part of the population, get any type of sample, whether it's a blood sample, a tissue sample, urine sample, etc. And you can really use any of these mediums to do analysis with mass spec. And what's beneficial about all of these methods, and specifically the substrate-based methods, so think about paper, thread, or even a cotton swab, you can do collection, storage, and mass spec analysis from one sample or from one substrate, which is great. We also can do solid and liquid samples. There's minimal to no sample preparation, but you get this ion generation from a sharp tip. So when we think about electrospray, an electrospray emitter has a sharp tip. If you do direct injection with a needle and syringe, your needle has a sharp tip. The substrate-based methods are no different. You have a tip of some sorts where you get your ion generation. And for most of the substrate-based ambient ionization techniques, we don't need gas, whereas for like desorption electrospray, you would need gas or things like that. So what we have been developing in my lab, part of what I have been doing is developing a technique called thread spray ionization. So this is just a very broad schematic of all of the applications that me and a colleague, Sierra Jackson, have been working on. We basically took the most common analytical applications and split them between each other and have been trying to develop this method. So I particularly have worked on illicit drug analysis. I've also done some environmental studies with ricin, and I've also been focused on these omics. So lipid and fatty acid analysis, but also metabolites for metabolomics. And essentially all thread spray is you take a single piece of thread, house it inside of a glass capillary, add solvent and voltage, and you get a generation of ions from the tip of the thread to do analysis. So we're going to talk about a little bit about my illicit drugs project. So for thread spray, like I said, you can couple LC to mass spec with either electrospray or APCI. I've been exploring both ESI-based and an APCI-based methodology for thread spray, which has been a lot of fun. So we're going to talk about the ESI-based one today, but in general, ESI is really good for high polarity analytes, whereas my APCI work is all with low polarity analytes, and they look the same. The only real difference between these two is the solvent that I use, really. And sometimes the voltage is a little different, but I tend to stay between 3 and 5 kilovolts anyway, like most ambient ionization techniques. So for this first project, what I was looking at was 3 different types of thread. So I use cotton thread. And when I mean thread, I mean exactly what you're thinking. Like I went to the fabric store, bought a spool of thread. The same spool of thread I bought my first semester of the group is still the same spool of thread that I use. So it's very cheap and it's also easily accessible for people. So I focused on 3 different types of cotton thread because we were really interested in seeing what surface effects could we manipulate with our platform. Is it really good for like with a hydrophilic surface, a hydrophobic surface, etc. So this untreated thread is just regular cotton, so it's completely hydrophilic. This 30-minute treated thread is this intermediate between hydrophilic and hydrophobic, and this 60-minute thread is completely hydrophobic. These are all tandem spectrum for cocaine. It's 100 ppb, both in regular solution, so in ethyl acetate, and then also in blood, which is what I focus on for this first project. So a few things should jump out at you. You should already notice that my non-biofluid samples have higher signal intensity than my biofluid samples. And that's what we expect because blood is a complex matrix. So we know that we're gonna get some form of ion suppression. Another thing that you should notice is that as we treat the thread and as it becomes more hydrophobic, we do get an increase by 1 order of magnitude for the tandem spectra for cocaine, which is really great. So I took advantage for this project of this increased ionization efficiency that the completely hydrophobic thread warrants. Now, this is not to say that the untreated thread is still not good. I still use the untreated thread, and the 30 minutes is a really good intermediate between the two surfaces. It's just cocaine, it works really well, and the hydrophobic thread does give you this extra level of sensitivity. So knowing that, my favorite application for this project was we mimicked tissue analysis. So with Dr. Amanda Hummond's lab here at Ohio State, they have agarose beads that they use for their a lot of their experiments. So she gave me a bunch of agarose beads to really just play with. So what I did was I wanted to see if I could use thread spray to sample these beads. So I looked at diazepam in these beads and I essentially just doped extra media that they gave me with diazepam. I removed the media that was already in the well plate when they gave them to me, added the doped media, let it infuse overnight and sampled it. And all sampling looked like was me taking a needle and thread and pushing it through. And these are just pictures of what it looks like when it was a viable bead versus when it was non-viable. So we only focused on viable beads for this particular part of the project. And as you can see, we get very reproducible diazepam tandem spectrum, confirming that thread spray can be used for quote unquote solid sample analysis. We didn't stop there. We wanted to see how stable it was. We wanted to see if we could force the beads to become non-viable. So like this day 21 picture is here. Could we still see signal for diazepam? Because we know that we get this extra ionization efficiency, which does aid in increased sensitivity. So we know we can see differences in low concentrations of diazepam, but can we still track diazepam even in the trace amounts? And as you can see from this graph on my right, that we can still see it. Even after the sample starts to degrade, it is still sensitive enough to detect or pick up those low concentrations. So hopefully I've been able to convince you that we can use thread for small molecule detection. The hydrophobic thread is really great because it does minimize these matrix effects and increases ionization, especially for blood samples. I can also control these extractions, which is really great without risking solvent or sample evaporation like other ambient ionization methods suffer from. We also can get this long-term analyte stability, which is really awesome. And I've been developing thread to be used as an all-in-one substrate. So without having to do extensive sample preparation, I can load the sample, store the sample, directly analyze both liquid and solid samples for small molecule detection for various applications. So I want to give a special thanks to my group. So this is my advisor, Dr. Badu Tuia. I want to thank Sierra Jackson, who's pictured right here, and my undergrads, Hannah and George, for helping me with a lot of this work, and the rest of my group members, also Ohio State and ASMS for funding for this particular project. If you want to keep up with me, you can email me. Here's my email, my professional website, and also my Twitter. But I want to thank you all for your attention, and thanks to FeMS for allowing me to talk about my research. And with that, I will take any questions.
Great job, Devin. So if you have any questions, please go ahead and unmute yourself. I have a quick one for you. In your cocaine analysis, I saw that you have the parent peak and then the minus OH peak. Which ones do you follow, or which ones do you look for to know that you have cocaine?
Yeah, so the cocaine transition that I look for is this 304 to 182. So that's the loss of benzoic acid.
Okay. Yeah, that makes sense.
Yeah.
There must be some other question out there.
I'll ask a question. So this is fascinating. I'm just trying to think of like crazy things that you could do with the technology. So I just got off a call where we're going to be tracking migrant workers to look at pesticides and their response to different aspects of their life, the environment. So could you, for example, use thread from their clothing to look at pesticides ultimately, or to look for other stuff? Like what kind of concentration of analyte do you need? I know that's kind of a stupid question because it all depends on the analyte, blah, 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 but how do you see this working in situations like that?
Yeah, so it's funny you asked that. So Sierra Jackson, who's the other person that does thread spray, she actually has a pesticide project, and she was looking at different pesticides, so like glyphosate, atrazine, et cetera, both in surface water, so in river water, but also on fruits, which is really interesting. So we can do that type of analysis. My stuff is more clinically focused. So I've done like environmental studies with ricin. I've done almost all the biofluid analysis that you kind of think of for various applications. But thread spray in general can warrant you on like the parts per trillion and I guess lower concentration ranges, which has been good, which really helps for trace analysis for almost all applications that people are actually interested in. So anything around like roughly, I think for all of this work for this first project, I stuck at 100 ppb for almost all of my experiments because I really didn't need anything super concentrated. So it is pretty sensitive.
Very nice. Yeah, nice.
I would ask, being a transition talk was there, and you said you were 5th year, right?
Yes.
Are you looking for next?
Yeah, technically I'm not looking for next anymore. And that happened like 2 weeks ago. So I'm actually actually accepted a position in industry. So I'm really excited about that.
Congratulations, that's fantastic.
Thank you.
Can we ask what you chose?
Oh, I'm gonna work for Merck as a senior scientist. Yeah.
Oh, okay. Maybe you said that and I just missed it. I'm sorry.
Oh, no, I definitely forgot to say that, so I'm glad you asked.
Well, congratulations. That's fantastic.
Thank you.
That is fantastic.
When will you start?
In June.
Oh, nice. Oh, that is awesome.
Thank you.
Well, if no other questions, let's give Devin a big round of applause again, and we'll hand over the reins to Erin.
All right, I will be your second talk from Ohio State today. So Devin and I both go to Ohio State, however, we're in different research groups. I'm in Vicky Wysocki's research group and also part of the P41 NIH National Resource for Native Mass Spectrometry Guided Structural Biology. And so today I'll be presenting my work on modifying our 15 Tesla ICR for native mass spectrometry applications. Okay. To give you a bit of personal background about me, I was actually born and raised in Ohio, probably 20 minutes outside of Cleveland. I graduated from Marietta College in 2013 with my Bachelor's of Science in Biochemistry. From there, I took 3 years off and worked in the pharmaceutical industry, both as a quality control chemist and then in HPLC method development chemist in Wilmington, North Carolina. And then I really wanted to get back into research and wanted to look at mass spectrometry because I had never used one before and wanted to get into instrumentation. And that brought me back to Ohio to join Vicky's group. And also just some personal information about me. I love watching soccer. I played soccer for my entire life and 4 years throughout college. And this is my niece and nephew. They are my new trainees. One of the good things about COVID was Ohio State shut down and I went to go stay with my parents and my sisters back up in Cleveland for 2 months. And so we had soccer camps where I trained them all day. So they're going to be my replacements and be my future soccer stars. So as far as my research goes at Ohio State, it's focused mainly on mass spectrometry development. And so I've broken that into 3 different categories. Instrument development, so taking commercial mass spectrometers and adapting those for native mass spectrometry applications, and specifically incorporating trapped ion mobility spectrometry, or TIMS, and surface-induced dissociation, SID, on our ICR platform. I've also looked at mass spectrometry fundamentals, so looking at the process of nanoelectrospray ionization and how we're transferring proteins and protein complexes into the gas phase. Additionally, I've looked at peptide fragmentation mechanisms using infrared spectroscopy. I've really enjoyed this project because I get to use a free electron laser coupled to a mass spectrometer. And so I either am going to Felix in the Netherlands or Clio just outside of Paris, France, to work on this project. And then also we look at how this technology and the understanding of these fundamentals can be applied to biological systems. So working on applications and collaboration projects. So working with Pfizer using SID to look at ligand binding. And now we're currently in a collaboration with the NOIC group at UC Irvine to study amyloid beta peptide aggregation using ion mobility. The bulk of my dissertation work has focused on instrument development, so that's what I'll be presenting to you today. And so we're using native mass spectrometry to study protein structure. And so protein structure can be classified into four main groups. So you have your primary structure, where that's your amino acids and your polypeptide chain. And those form localized structures such as alpha helices or beta sheets. The overall fold of a protein is considered the tertiary structure. However, in order to carry out protein function, most proteins exist in what we call complexes. So either protein-protein interactions or protein-ligand interactions. And these ligands can be DNA, RNA, or lipids, for example. However, we're not just interested in taking a snapshot of the protein structure, because we know that proteins are often dynamic structures. They exist in different conformations and they interact with molecular species to carry out their function. So we need to develop technology that can capture those aspects. And so that's why native mass spectrometry is advantageous. However, most commercial mass spectrometers are geared towards low M/Z analysis or small molecule analysis, specifically the ICR platform. But we want to take advantage of using an ultra high resolution and ultra high mass accuracy platform, so we can study small mass differences and also identify species that overlap in nominal M/Z. And so if you're unfamiliar with the Bruker ICR, this is just a basic instrument diagram. We're generating our ions with either ESI or nano ESI. We have this entrance funnel region, which focuses and desolvates our ion. And then we have a mass selection quadrupole, a collision cell to carry out CID and accumulate ions before we pulse them into the ICR cell. And so the first instrument modification we performed was replacing this standard entrance funnel region with a trapped ion mobility spectrometry funnel to perform ion mobility. And then we also replaced the front end cap of the collision cell to include a surface-induced dissociation device. And it is important to note that our ICR is a shared user facility instrument. It's actually part of the Ohio State Campus Chemical Instrument Center. And so there's multiple users on this instrument. So we're doing native. Our pod, who I'll show you on my acknowledgement side, is doing small molecule MALDI analysis. And then also Amanda Hummon's group at Ohio State is using this instrument for MALDI imaging. And so any modifications that we're performing cannot compromise the performance of the instrument. And they also need to be easy to either take in or out, or ideally leave in the entire time. And so we worked on implementing trapped ion mobility spectrometry, or TIMS. And there's really 3 steps to performing a TIMS analysis. And so the first step is accumulating our ions. So we're generating those for native with nano ESI, and we have this deflector plate. And so we'll set that to a relatively repulsive voltage to focus our ions into the TIMS funnel. And then we're trapping the ions. This is where we perform the actual mobility separation. So we then change this deflector plate to be a relatively attractive voltage. And so we're only doing mobility separation and no longer sending ions into the ion mobility funnel. And so we apply an electric field gradient, and based on an ion's mobility, they'll be trapped at a certain location on this electric field gradient. And finally, we'll elute those ions out of the TIMS funnel. And we do that by, in the stepwise function, lowering that electric field gradient until ions are no longer trapped. And so to give an example of improved resolution with TIMS, this is looking at the bradykinin peptide. And we have DESR1 and DESR9 as purchased from Sigma Aldrich. And what that is you can either delete the N-terminal arginine or the C-terminal arginine. And so these peptides have the same nominal mass to charge. So how can we separate them? We can't use a traditional analytical quadrupole. And previous work in our group utilized traveling wave ion mobility on our water synapse platforms. And you can see we can separate out and identify the different forms. However, with trapped ion mobility spectrometry, it is a higher resolution ion mobility separation because we're separating species based on that electric field gradient. And so we can get very nice separation and almost baseline resolution of these two overlapping species in M/Z. The second instrument modification we performed was adding SID capabilities to our ICR and improving the existing technology. And so a brief background in tandem mass spectrometry. Most commercial mass spectrometers come equipped with collision-induced dissociation, or CID. This is very efficient for small molecules such as peptides. However, when you do CID of protein complexes, you tend to produce a very highly charged monomer that's indicative of unfolding and the corresponding N-1 mer. Compared to surface-induced dissociation, which is what the Wysocki group develops, where you accelerate your ion of interest into a rigid surface, and you tend to produce compact subunits that are indicative of the native structure. And so this is a single high energy deposition step. And so you can access dissociation pathways that you can't with CID. And so here with this example, I'm showing a dimer of dimers. And so with SID, we cleave the weakest interface to produce those two dimer subunits. And so we can get information on how protein complexes are natively structured with regards to their connectivity, stoichiometry, topology. We can probe gas phase restructuring. And also with SID, you can probe subunit ligand binding and interactions, which with CID, you typically see ligand loss because you're undergoing numerous low energy collisions. And so for the SID on the ICR, when I first joined the group, an outgoing student, Jing, had developed the Generation 1 device, and we've named these in terms of when they've been developed. So Gen 1 was the initial device design, and it consisted of 10 electrodes that are independently controlled with an external power supply with the DC power. And we truncated the original collision cell that Bruker provided. And so one of the main critiques of this device was having to tune 10 individual lenses for people who don't work with this device regularly. They found it very challenging and very complicated. And so we wanted to simplify it. Additionally, by truncating this collision cell, we had a diminishing performance in CID, especially for small molecules. And so because this is a shared instrument, whenever we wanted to perform SID, we would have to vent the collision cell chamber, put this device in, and then when we were finished, take it out. And venting the instrument and pumping back down would waste about a day or a day and a half. And so we wanted to not only make this a simpler device, but also more efficient, and also not diminish CID performance for our multiple users. And so we started eliminating electrodes. So we then came out with Gen 2, in which we simplified the number of electrodes. And then even farther from there, we came out with Gen 3, which is an order of magnitude smaller. And so this work was spearheaded by Dalton Snyder, who's a research associate as part of our P41 facility, and has served as a wonderful mentor to me, and I've really enjoyed working with him. And so this Gen 3 SID device replaced the front end cap of the Bruker commercial collision cell, and it's a 3-lens device. And so we have one half of this lens is the surface. So actually in the aperture is where the ions are hitting. And then we have a second lens, which is the deflector, which we tune to guide ions into the surface or accelerate them into that surface. There's a peak spacer, and then finally an extractor lens at the back, which we use to guide ions off of the surface and to downstream optics. And so to show an example of combining these two technologies, I'll show streptavidin, which is a 53 kilodalton homotetramer. And so this is the mobility distribution here, and we get quite nice separation of the 12 plus, 13 plus, and 14 plus tetramer. And so you can actually slice this mobility and do what we call mobility parking on that specific charge state and perform SID. So here I'm showing Tim's selection of the 13 plus precursor and performing SID at 520, 780, and 1040 electron volts. And a lot of my work went into figuring out how to optimize signal to noise and trapping of protein complexes. And so here I'm showing suitable signal to noise. We also had an issue of having an unknown reaction occurring in our collision cell. And so once we identified that, we cleaned every single part of our collision cell and also switched out our gas lines. We got rid of that reaction. And because we're working on an ultra high resolution platform, we can zoom in on overlapping oligomers. So here I'm producing dimer 6+ and monomer 3+. But if we zoom in, because we're on an ICR with ultra high resolution, we can actually identify and relatively quantify those overlapping species. So these blue dots here are showing dimer and the red dots are showing monomer. And so at low SID energies, we're producing predominantly dimers, but then as we increase the energy, we're further fragmenting to produce predominantly monomers. So we can take this information and look at how the native structure is put together. A second example is looking at cholera toxin B, CTB, with GM1S. And this is a pentamer, and it binds 1 GM1S, the sugar head group, per subunit. And so the pentamer binds 5 total subunits. Again, we're looking at the mobility distribution here. So we have the pentamer 13 plus, 14 plus, and 15 plus. And we can take a slice of the 14 plus. And here I'm showing the TIM selection with CID or TIM's SID. And just looking at the mass spectra, you can see a very clear difference. And if you look over here at the deconvoluted mass spectra, so correcting for charge and only looking at the mass domain, TIM CID produces predominantly monomer and the corresponding tetramer with ligand loss. So we see tetramer with 5 GM1S, 4, and 3. Whereas with SID, we're seeing monomer, dimer, trimer, and tetramer products with either 0, 1, or 2 GM1S, and dimer with 1, 2, or 3. And this can provide information on our dissociation pathways to then look back at how these ligand interactions are occurring. So with CID, we take our ligand-bound pentamer and we only see highly charged monomer with tetramer and ligand loss. But when we look at SID, we're seeing a range of different dissociation products. So for example, we see monomer with no ligand, with one ligand, or with two ligand. And this confirms that the ligand is binding between the subunits as opposed to being embedded within a single subunit. And so from my instrumentation work, I've optimized TIMs for trapping of large protein complexes. This was a technology initially developed for small molecules. And so they kind of told me, hey, good luck, try proteins and protein complexes. And so we did that. We developed user-friendly, more efficient SID devices. And there seems to be some good excitement around this Gen 3 device. Dalton has also installed it on multiple flat platforms, including Orbitraps and QTOF, Water Synapse. We've also combined TIM's SID technology together for protein complexes. And for future directions, we are in an active collaboration with Bruker developing their timsTOF Pro for native MS. This work was done on a 15 Tesla ICR, which we are very fortunate to have, but not many research groups do have. So we're hoping by developing some of this technology on the timsTOF Pro, it's on a more accessible instrument and can reach more research groups in the future. And with that, I would like to thank the Wysocki Research Group. This is Vicky in the back, there's me. This is Dalton, who I mentioned before, who has worked a lot with me and been instrumental in my development as a graduate student. Also Arpad, I call him my Mass Spec best friend. He works in our Campus Chemical Instrument Center, and he also works on the IRMPD project with me. So we usually travel to Europe every summer and get into trouble, but we have a great time. Our collaborators at Bruker, Mel Park and Mark Ridgway, of course, they've helped us develop the TIMS technology. And also Desmond Kaplan from Cap Science. I didn't get to talk about it today, but we did develop a low frequency RF driver for our quadrupole on the ICR to do high mass selection. And he's also just been a great resource to bounce ideas off of. I'm also financially supported by the Ohio State Sesquicentennial Student Leadership Scholarship. And also I wanted to give a shout out to the Pod 14 FeMS mentorship group. We've named ourselves Taking the Leap because we're all in transition periods. And this work was financially supported by NIH, specifically our P41 resource for native mass spectrometry. We have a website if you have any biological questions that you thought could be answered with ion mobility or SID. It's not limited to our ICR. We have several students working on Orbitrap and ion water synapse instruments with different forms of ion mobility, SID, and UVPD. We also have a Twitter as well. And with that, I'd be happy to take any questions.
Oh, that was great, Erin. So if anybody has any questions, please unmute yourself. So I guess to start off, it seems like the reaction pathway for SID would be really complex to, I guess, manually understand. So are you guys working on machine learning to kind of deconvolve the abundances from the different complexes you get out of it?
So it really depends on the structure of the precursor. So something like streptavidin that I've shown that we know as a dimer of dimers, we can really predict the fragmentation pathways that it will follow. Something for CTB, which is cyclic, it's a pentamer, so it has equal interfaces between each subunit. So it produces monomer, dimer, trimer, tetramer pathways. That is complex. Typically, we do use some deconvolution software. A lot of us use Unideck from Michael Marty's group. So that's the common software that we use for deconvolution currently.
Nice.
And he's also one of my Pod 14 mentors, so I have to that plug in.
That's perfect. Other questions? Everybody's so quiet tonight. Go ahead, Amber.
Sorry, I double muted myself. I would ask the same question. If you alluded to that your pod is all in transition, so it sounds like you are too.
Oh yeah, I forgot to mention that. I'm in my 5th and final year. I'm in the process of interviewing with 2 positions. One is biopharmaceutical and one is an instrumentation position. They are completely different, and I honestly couldn't tell you what I want to do with my life. I'm just kind of figuring it out as I go. It's really research driven as opposed to position. So it's whatever they're working on. If I find it stimulating, I'm all for that.
Yeah, so I think the big thing to realize is that whatever decision you make now, it's not for life. I mean, you can change your mind. So I think your attitude is great to take whichever one interests you most and make the most of that job and see where life takes you, right? I read somewhere that an average person has something like 12 or 20 jobs in their lives, different jobs.
Well,
Yeah,
then
I was going to say
I've got room to go.
yeah, yeah, I was going to say if you figured out your life, you need to help me.
Could do a consulting business.
Yeah.
Life coach.
I'll give Erin a round of applause then. And the great talks, both Devin and Erin. I really enjoyed both of them. I learned so much from the talks. So to end up, let's do our normal break.